you a study of TRIM21 functions as an oncogen in glioblastoma by transactivating FOSL1 and promoting the ubiquitination of B27.
Glioblastoma, type of malignant and aggressive brain tumor that originates glial cells in the central nervous system. Oncogen, gene that has the potential to cause cancer. TRIM21, immune system protein that recognizes antibody virus complexes inside the cell and directs them to their degradation through ubiquitin. B27, cells cytokine inhibitory protein that stops cell proliferation and function as a tumor suppression. FOS1, transcription factor of the AP1 complex that regulates cell proliferation and survival genes.
TRIM21 action. So first we have the degradation of tumor suppressor B27. So TRIM21 facilitates the degradation of this protein via the ubiquitin proteasome pathway. This degradation accelerates the G1 to S phase transition of the cell cycle. Furthermore, it upregulates glioblastoma stem cell markers such as these two, thereby promoting the cell renewal capacity of tumor cells. This specific function of TRIM21 is mediated by its ring domain. Then we have the transactivation of oncogen FOLS1. So TRIM21 physically interacts with FOLS1 and oncogen. These interactions enhances FOLS1 self-transactivation. FOLS1 activation in turn further increases the expression of these two markers, contributing to glioblastoma tumorigenesis. This function of TRIM21 is primarily mediated by its price-right domain.
General objective. This study investigated the role of TRIM21 in glioblastoma, including its interaction with FOLS1, its regulation of FOLS1 transactivation, and its ubiquitination mediated degradation of tumor suppression B27.
Métodos prueba PCR. Propósito general. Se utiliza para medir y cuantificar los niveles de expresión de genes específicos a partir de una muestra de ARN. La aplicación en el estudio se empleó para evaluar si los cambios en niveles de la proteína P27 observados al manipular TRIM21 se debían a una regulación a nivel de ARN mensajero. Western blot. Propósito general. Técnica para detectar y cuantificar proteínas específicas en una muestra biológica. Permite analizar los niveles de expresión de proteínas, su tamaño y ciertas modificaciones posttraduccionales. Se utilizó para cuantificar los niveles de expresión de TRIM21, FOLS1 y P27 en diferentes líneas celulares de glioblastoma.
La citometría de flujo es una técnica de laboratorio que analiza simultáneamente las características físicas y químicas de múltiples células individuales a medida que fluyen a través de un haz de luz. Permite medir el tamaño, la granularidad y la fluorescencia en cada célula. La inmunohistoquímica es un método de laboratorio para el cual se usan anticuerpos con el fin de determinar si hay ciertos antígenos o marcadores en una muestra de tejido. Por lo general, los anticuerpos se unen a una enzima o tinte fluorescente. En el estudio se utilizó para evaluar la expresión del TRIM21 y su asociación al FOLS1, confirmando los niveles de TRIM21 en los gliomas.
Results Figure 1. In this, we know the interaction between TRIM21 and different cell lines, specifically in these ones. Then we know the interaction between TRIM21 and FOLS1 in this type of cells, and the same with this one. In this immunofluorescence, we know the interaction between FOLS1 and TRIM21 with different cells, as they were colocalized in the nucleus. And then we have this direct interaction between FOLS1 and TRIM21 was detected by GST pull-down assay. Figure 3. In this one shows that B27 protein levels are regulated in glioblastoma cells, which silence TRIM21 compared to control cells. In this one, glioblastoma cell lines expressing TRIM21 were treated with the proteasome inhibitor MG132 shown here. And in this last one shows that P27 protein levels remain significantly more stable in silent stream 21 cells compared to silence control glioblastoma cells.
Número 4. En las líneas celulares del glioblastoma U87MG y HTV15 se silenció el ITRIN21, que hace referencia a este. Y se analizó la ubiquitinación del P27 después de la inmunoprecipitación del P27. Y se comparó con el control silenciado, que hace referencia a este. Al número 10 podemos encontrar que en los paneles del A al D se muestran las secuencias de sitios de unión de APN promotor comparado con el wild type versus los mutantes. En el panel E, el Western plot confirma la sobreexpresión del TRIP21 en U87MG y HTV15. De los paneles de la F a la G, en las células con TRIP21, la actividad promotora wild type de AP21 aumenta y cuando se introducen mutaciones, la actividad TRIP21 disminuye notablemente.
In the description, these authors said nuclear localized TRIP21 drives tumorigenesis by promoting FOLS1 transactivation via AP1 binding sites. This agrees with the article. Then, these authors said TRIP21 facilitates dose-dependent degradation of P27 through ubiquitination. This agrees with the article. Then, these authors said in nasopharyngeal carcinoma, TRIP21 ubiquitinates and degrades tumor suppressor. PHD1 following this, downregulation enhancing NFKB and STAT3 activity, which promote cell proliferation and tumor progression. This agrees with the article. And then these authors said TRIP21 physically interacts with fossil 1 and P27. This agrees with the article.
Conclusion. The first one. TRIP21 serves as the therapeutic biomarker and diagnostic of patients with glioma. This discovery was made possible thanks to the different laboratory techniques that molecular biology implements in the medical practice. FOLS1 works as an oncogen. This promotes the growth, invasion, and the pluripotency of the cancer in glioblastoma cells. Molecular biology contributes to the study of behaviors and functioning of different microorganisms, achieve the development of the effective treatments. Molecular biology implements different laboratory techniques that contribute to the investigation of processes that may or may not favor pathology.